A very good morning to all of you. So students, here we are taking up another topic in the circulatory systems, which is the evolution of heart. So this particular topic is very important as per your examinations are concerned. So you can get a full-fledged question on this particular thing, which we call it as evolution of heart. Now, before starting this particular topic, I must give you some concepts of for the basis of that how we take the evolution of the heart in different animals. Now, first of all, if we start with the primitive animals, suppose we had the protozoans. They don't have any kind of like multicellular body, so they are unicellular animals. So they are not requiring any kind of a particular system, but they had a kind of cyclosis or the streaming movements of the cytoplasm. So that was their mechanism of circulation. Now, first of all, we have to take the concept that why we have this particular system in our bodies. So the major, you can say, the function of this particular system is to transport the things from one place to the other place. So as the animals, they grow in size and efficiency and mechanisms, so we have various types of circulatory systems developed in different kinds of animals. So with the evolution in the environmental conditions, or you can say the evolution in the morphological conditions or the physiological mechanisms, so all these systems, they have changed in due course of time in evolution. So if we take some further animals, if we take poriferans, they have got their own canal systems for the circulation of extracellular fluid. So we had taken up in the previous topics that how the circulation of the fluid, it takes place in the coelentrates even. And here we had seen that these animals, they have got the gastrovascular cavities. So these gastrovascular cavities, they are filled with the gastrovascular fluid, and that is the coelomic fluid in the coelentrates. So that was transporting the digested food along with the other materials, the ions or the salts, to the various cells of the body. So if we go further, suppose we have the higher animals, as in the case of platyhelminthes, or you can say the annelid phylum. So this particular phylum, the Annelida, it was the first phylum to have some kind of vessels which are dedicated for the circulatory systems. If we take the prior phylum, first of all, the Aschelminthes, so those were roundworms, but they were not metamerically segmented. So they had a coelom inside their bodies, though it was a pseudocoelom, but they have got pseudocoelomic fluid. So that particular fluid is working as a circulatory system in those animals. Whole of the food which is taken in the mouth and then it goes to the intestine, as they had very simple systems, and we had discussed the system of the Ascaris. Must have gone through Ascaris in BSCs. So Ascaris, it had very simple kind of systems, and when the food reaches the intestine of the roundworms, it gets absorbed into the coelomic fluid, and that coelomic fluid is continuous throughout the body, and all the substances can be transported from one place to the other. In case of earthworms, or you can say annelids, typically, we had a typical circulatory system developed for the very first time in the evolution of the animals. Now, here we had five longitudinal vessels. So, if you remember the circulatory system of the earthworm, we had a dorsal vessel, we have a ventral vessel, and so many other longitudinal vessels. I am not going much into that particular topic, but still, I want to say is that these animals, they have got long vessels throughout the body, and out of these the main important thing was that the dorsal vessel of the annelids, that was pulsatile in nature. So that was the basis, that was the origin of the pulsatile ability, you can say, in the vessels. So in that particular case, if you take the case of earthworm, the dorsal vessel of the earthworm is pulsatile, and it is bringing the blood from the hind end towards the interior end of the earthworm. So that means there is a single direction of the flow of the blood, and it is very, very important for the heart. Number two, the heart should be pulsatile. That means it should be rhythmically contracting under some pacemaker system. So that vessel in the earthworm was also pulsatile. It was also taking the blood in a particular direction. The third major thing in that particular vessel was that it was having valves. So this is a third major characteristic of any heart, that it should have valves. So if the valves were present in the dorsal vessel of the earthworm, that were for the checking of the backflow of the blood in that particular vessel. So once the blood has moved towards the anterior side, it cannot go backwards. So that is the major role of the valves in that particular animal. Now, if I go further, in case of earthworms, we had 8 hearts. So there were 4 pairs of hearts. And in BSc, I think you must have gone through these types of hearts. So first type was the lateral esophageal hearts, and the second type was the lateral hearts. So these are the two categories of the hearts in earthworm. So those were having valves. They were single directional. So blood was moving, I should say, the blood was moving from the dorsal vessel to the ventral vessel in those hearts. Anyhow, that was the case of the earthworm. So, going briefly to this topic, I want to say that the earthworms or the annelids, they have developed the first circulatory system which is typical in nature, and it was quite efficient to produce specific, you can say, blood pressure to maintain the whole body of the earthworm. Now, if we go further, if we go for echinoderms, we have water vascular systems. So you have gone through the water vascular systems of the echinoderms. So you must have gone through the starfish in your BSc. So after this, the vessels, they were, you can say, those were enlarged in size to some extent. They were ampulla-like. They are making some bag-like structures. And they developed a special muscular tissue. And ultimately, those bag-like structures were considered to be as the hearts of the animals. So that was a due course of evolution of the hearts. And if we take some further examples of the urochordates and the protochordates or cephalochordates, so you might find some types of the hearts in those animals also. So I'll be taking some examples in this particular lecture. for the evolution of heart. So what I want to say, as I started from the protozoans up till now, what is the basic need of this particular system, or why we need hearts? The heart is a pumping organ which can transport an adequate amount of the blood to all the parts of the body. So this is a major role of the heart. So as the environmental conditions of the animals, they changed, the size of the animals, it changed, the requirements of all the animals, they changed. So the heart also changed with time. So this is a course of, you can say, advancements of the heart with the environmental conditions and the time periods. So that is the evolution of the heart. So the basic need of the evolution of the heart was to cope up with the different or changing environmental and morphological conditions of the body. So students, in this particular topic, I'll be taking various examples, and I'll be starting this particular topic in case of vertebrates. Now you can get a question, full-fledged question on the evolution of heart in case of vertebrates. So now I'm going to start the particular topic, and every single animal is needing hearts to pump the vascular fluid, which we call it as blood, to the different parts of the body to maintain a particular levels of oxygen and food supply. So this is a basic need of the heart. That's why we need the heart. Okay, so if I go further in this particular lecture, first of all, we have the example of the Amphioxus. Now, this particular animal is having various gill slits. So I'll be showing you these gill slits. So in the diagram also, you can very well see this is the pharyngeal region of the Amphioxus, and a number of gill slits, can be like 60 pairs. They are present in this particular animal. So this is the particular seat of oxygenation in this particular animal. This part, the red part, is the seat of oxygenation. Now, if I give you the idea of the heart in this particular animal, what we have is, we have this kind of pharyngeal region of Amphioxus. Now, this particular diagram is showing the arrangement of the aortic arches in case of Amphioxus. So here you can very well see, this is a very important thing to note, that at the very top, we have got two dorsal aortae. So these are called as paired dorsal aortae. And when they go back towards the posterior end of the body, they meet together and they form a single dorsal aorta. So they are joining on the posterior side of the animal. Now here in the pharyngeal region, they are two in number, and they are termed as paired dorsal aortae. Now we have a single ventral aorta on the lower side. So what is happening, that the ventral aorta is acting here as the heart of the animal. So here you can very well see it has been written that this is the heart, and ventral aorta is the heart of the animal. So there is no specific heart in the case of Amphioxus. And this ventral aorta is somewhat pulsatile to some extent, but that is not rhythmically perfect kind of a vessel as we have in the case of the earthworm, as we have discussed previously. But here also we have the tubular kind of heart in this Amphioxus, as it is a tube-like ventral aorta. Now, a very important thing, which is present over here, is the next part, which we call it as the bulbulus. Now students, this is the most important thing which I should discuss over here. See, these are the gill slits, and what is happening that the blood, it is coming into this ventral aorta in one direction. Now this blood, it goes into this bulbulus, and this bulbulus, it contracts to some extent. It is a bag-like structure. It is a pouch-like structure, the bulbulus. Now when the bulbulus contracts, the blood goes, it flows into the afferent branchial artery. So this black colored artery is the afferent branchial artery. Now the blood, when it is flowing through this afferent branchial artery, it is going towards this region which is the seat of the oxygenation. So that means in the ventral aorta we have the deoxygenated blood. Now here I should take blue line. So it is the deoxygenated blood and it is going towards this side. Then it rises up and it is going for the oxygenation process in the gill slits. So when it follows the gill slits, what is happening? Oxygenation occurs and this blood gets oxygenated. It goes from the efferent branchial artery into the paired dorsal aorta. That means the paired dorsal aorta, they had the oxygenated blood. The ventral aorta had the deoxygenated blood. Now this blood will flow towards the head side and towards the posterior side. So that will go to the head and the body parts of the amphioxus. Now here what is important thing is that which is the main organ for the pressure building. So here this is the bulbulus. Now these are the bulbuli, these are the pouch-like structures who are making the pressure, and thus the blood is going in the upward direction and it is flowing through the gill slits and then it is oxygenated and then it reaches the dorsal aorta, the pure dorsal aorta. So that means here the heart is the ventral aorta and it is aided by the bulbuli. So that is the case of amphioxus. So here we can have a huge number of the gill slits. We have got huge number of the efferent branchial arteries and we have got huge number of the bulbuli. So this is the case of amphioxus. Now students, after this we have the case of the cyclostomes. Now if we take the case of cyclostomes, these are very comparable to the fishes as per their hearts are concerned. Now this is a particular heart of a cyclostome and it is basically two-chambered. And now we'll be seeing that how this heart is two-chambered and what are the names of the chambers, what is the main structure of this chamber and how it is working. Now if I go further, this is the first chamber of the heart which is called as atrium. So this is the first chamber. Now if we go further, we have a second chamber which is called as the ventricle. So we also have auricles and ventricles, but here we have only one atrium or auricle, one ventricle. And here you can very well see that the atrium is bigger in size than the ventricle. And number two, which is very important thing to note, that the wall of the ventricle is thicker than the wall of the auricle. Now, why is it so? Because the ventricle has to pump the blood, it should be more muscular, it should be capable to produce more of the pressure when the blood flows through it. So this is the basic mechanism of the ventricle contractions. So it is having a thick muscular wall as compared to the auricle. So here auricle, it is just giving the supply of the blood to the ventricle, and this is the main part of the atrium. Perhaps that's why the atrium size is larger than the ventricle. So if we go further, we have an accessory chamber. Now this is not the actual chamber of the heart. This is not the true chamber of the heart. This is the accessory chamber of the heart of the cyclostomes, and this is called as sinus venosus. Now this chamber, it is the seat of storage of blood. Now what happens here, the blood comes from the body, it reaches the sinus venosus, and it is stored over there. Now this is a kind of chamber which can store the blood which is coming from the body for some time. And then you can very well see that here are the sinoatrial valves. Now these are the valves which are present in between the sinus venosus and the atrium. So these are called as sino for sinus, atrial for atrium valves. And we have another valve which is called as the atrioventricular valve, which is present in between the atrium and the ventricle. And why these valves are present? These are present to check the backflow of the blood. So once the blood it comes into the sinus venosus, it goes to the atrium. Now it cannot come back. It can go to the ventricle, but it cannot come back to the sinus venosus because of the presence of the sinoatrial valves. And once the blood flows into the ventricle, it cannot come back to the atrium because of the presence of the atrioventricular valves. So this is a single way of blood flow. So blood will be flowing from sinus venosus to atrium to ventricle, and then there is a fourth accessory structure which is present in this heart, and this is the conus arteriosus. Now students, this is again a very important structure which is present over here. This is a pipe-like structure with some valves inside it. So here we have the semilunar valves present in this particular structure. And the number of the valves, it is different in different animals. So they are many in number. And what is the main function of the conus? Conus arteriosus, basically it exerts a kind of pressure on the blood when it leaves the heart. So suppose if you have a pipe and you are watering the plants in the garden, and if you press the pipe to some extent, what will happen? So the flow of the pressure will increase, and the pressure inside the pipe will increase, and that water, it will be thrown away from the pipe to larger distances as the pressure increases. So this is the same function it is played by the conus arteriosus here in this particular heart. So when the blood flows from the ventricle to the aorta, it will be with the pressure. So this pressure is exerted by the conus arteriosus and the walls of the ventricle. So this is a pressure building mechanism which is uh, derived from the ventricle and the conus arteriosus. Now, once the blood flows from this particular heart and it is going out, it flows into the ventral aorta. So the aorta which is leaving the heart is the ventral aorta. It is ventral in position. Now this is very important thing to note. So the heart will be ventral in position also. That's why it is the ventral aorta. And in these types of hearts, what we have, this is a single flow of the blood. This is called as a single circulation heart. We have a double circulation heart. So in this heart, sometimes these hearts are also referred to as the venous hearts. Why they are called as venous hearts? Because this heart will always contain a deoxygenated blood. It will never get the oxygenated blood in whole of the life of the animal. So that's why they are referred to as the venous hearts uh, or a single circulation heart. So the deoxygenated blood from the body will be coming to the sinus gnosis, and it will enter the atrium to the ventricle through the conus. It will go to the ventral aorta, and this deoxygenated blood will go from the ventral aorta to the site of oxygenation. So it will be oxygenated first of all at the site of some gills or lungs. So here these are the gills. So when they are oxygenated, when the blood is oxygenated, then it will move to the body. Oxygenated blood goes to the body. It is being used by the body. When it is being used, it is converted into deoxygenated blood, and it is coming back to the heart again into the ventral aorta. Oxygenation to the body. From the body, it becomes deoxygenated. It is coming back to the heart. So that means the heart is always having the deoxygenated blood. That's why they are referred to as the venous hearts. Okay, students. So if we go further in cyclostomes, what we have, it is a two-chambered heart. So we can very well see that it has got atrium, it has got ventricle. So we are not taking the sinus venosus and the conus arteriosus as the chambers of the heart. It has got the atrioventricular valves in between the atrium and the ventricle. It has got smaller ventricle. So we all know that the ventricle is smaller than the atrium. Muscular ventricle. So here I have already told you why it is muscular, because it has to exert pressure on the blood to go into the ventral aorta. So that's why we have a muscular ventricle as compared to the auricle. The ventricle has the Cordy Tendineae. Now this is an important point. Our heart is also having the Cordy Tendineae. Now what are Cordy Tendineae actually? These are some you can say connective tissue kind of a thing, fibrous tissue, which is you can say these are helping the heart or the ventricle specifically, because they are present in the ventricle. So they are not allowing the ventricle to collapse. So the ventricle is altogether stretched due to the presence of the Cordy Tendineae. And when the ventricle exerts a pressure to contract or to expel the blood out, these Cordy Tendineae, they are also helping in exerting the pressure. So these are present in the ventricle of the cyclostomes. Now, The sinus gnosis, which is present over here, this is an accessory chamber. Now, it is receiving the blood from. Now, from where it is getting the blood? Now, this is the main point. So, it is receiving from three basic things. Number A, it is the common cardinal vein. This is the first point from where the blood is coming into the sinus gnosis. Now, what is common cardinal? This is a vein, this is a single vein which is coming in the center of the body, and it is taking whole of the blood from the organs to the heart. So this is the main vein which is coming from the body organs to the heart. So that's why it is the common cardinal. Next is it is the inferior jugular vein. So I think you should be knowing about jugular veins. These veins are coming from the head, from the brain. So they are taking the blood from the brain to the heart. So these are the jugular veins. Third is the hepatic vein. So it is coming from the liver. So some animals, they have the hepatic portal systems. So this hepatic vein will be coming from the liver to the heart, and it will be giving you the detoxified blood into the heart. So these are the three major veins from which the sinus gnosis is receiving the blood. The common cardinal from the body, jugular from the head, hepatic vein from the liver. Now students, this particular diagram is showing you the path of the circulation of the blood in case of the cyclostomes. So this is the example of the lamprey, and here we start with you have a ventricle. This is the ventricle. It is muscular, you can very well see it has got a muscular valve, and this is the atrium. This is the first chamber, this is the second chamber. So we have got a two-chambered heart, we have got a sinus gnosis, we have got a conus arteriosus, so that we have seen in the previous slide. Now what happens that the blood, it is going into the ventral aorta. So why I have taken a blue color for this? Because this is the deoxygenated blood. Now it has to go to some oxygenation place. Now where it is going? It is going to the gill capillaries. It is going to the gill level for oxygenation. So in this part, the oxygenation happens and the blood becomes oxygenated, and it is going to the dorsal aorta after the oxygenation. So this is the dorsal aorta on the very top. From the dorsal aorta, this oxygenated blood is going to the different organs of the body. So this is going to the body capillaries, to the tissues, to the organs, and now the oxygen is being used by the body organs. So what will happen, that a deoxygenated blood will return from the body to the heart. So it is coming back to the heart. So this will be the deoxygenated blood. So this is the course of circulation in case of the cyclostomes. Now students, we have the case of the fishes. Now this is a very similar kind of situation in the fishes also, as we have seen in the case of cyclostomes. So we'll be taking this particular lecture